Hi, in this video I have prepared two questions for you. One is multiple choice question, another one is true-false question. And as usual, I recommend you to stop video here, try to solve both questions on your own first, and when you would be ready you can run video again and you can compare your answer with my answer and explanation. So here's the first question. If the sequence of an RNA molecule is as follows, what is the sequence of the template strand of the DNA? And in order to solve this problem, you need to know the names of the DNA strands. So imagine that here is a process of production or transcription of the message RNA, and here we have two DNA strands that separate, and here is the message RNA. So this is M, R, N, A, and this two strands is DNA strands. So this strand here that serve as a template is called template strand of the DNA. So template. And of course the other strand would be non-template. But the problem is that in many textbooks you may find different names for these two strands of the DNA, and for example the strand that is here on the top would be the code of this sequence would be the same as message RNA. That's why this strand of the DNA also called coding strand. So coding strand and non-coding. And also strand of the DNA here on the top called as positive strand, and this strand below called negative strand, or plus and minus strand. So as you see, according to our problem, we have to find the sequence of the template strand of the DNA. So this strand of the DNA. And we can find this easily. We just have to know the simple rule that A or adenine always pairs with thymine and cytosine with guanine. And in our example we have message RNA, so here instead of thymine we have uracil, so we can also say that A pairs with thymine in DNA or uracil in message RNA. So now we can easily build a sequence for the other strand of the DNA. So here we would have C here, G here, and T here, A here, G, C, T, and G, and C. And as you see, we have two such sequences here, and this is the first one sequence, as you see, CCGTA, so the same sequence. And here we have also the same sequence as this one, so which one to choose? And here's another rule. As you know, strands of the DNA goes in reverse order. So in order to assign ends here, you have to know that message RNA always transcribed with 5' end. So as you see here, the transcription started here and goes in this direction, and the first loose end would be 5' end. So now we can assign all the ends to the DNA strand. So this is going to be 3' end here, because this message RNA strand and this DNA strand would go in reverse order, and this strand of the DNA here on the top, that is coding strand of the DNA, would have the same prime ends as message RNA. So here we would have 5' end, here we would have 3' end, 5' end here, and 3' end here. So as you see, coding strand of the DNA goes in the same direction as message RNA. So now we can assign prime ends here. So this is going to be 3' end here and 5' end here, because we have template strand of the DNA. So if we have 5' end here on the message RNA, we would have 3' end here, and 3' end here on the DNA strand, and 5' end here on the message RNA strand. So here is our sequence, and as you see now, the correct answer would be answer D. So next question or statement: a eukaryotic promoter contains a Pribnow box. Is it true or false? And this is false. What is a Pribnow box? Pribnow box sequence is TATAAT, and we can find this sequence in prokaryotes, in bacteria, and eukaryotes. As you see in our statement, we have eukaryotes have a different sequence or different box, that is Tata box. This is a consensus sequence, meaning that sometimes here can be different base, but most frequent sequence would be as follows. And if our question about Pribnow box, this is sequence of the Pribnow box, and also this is called -10 region, and we can find this region 10 nucleotide bases upstream from the start codon, that is AUG. So we call this sequence in the eukaryotic organism as Tata box, and we call this sequence, that is TATAAT or Pribnow box or -10 region in prokaryotes. So RNA polymerase attached to this sequence, and this is where two strands of the DNA separates, and as you see, this fragment rich with T and A, thymine and adenine, and why? Because these two bases make double bond, and cytosine and guanine make triple bond. So of course it would be easier to separate this couple of bases than this couple of bases, and if we consider this region would have 12 bonds, and if we consider this couple, if we change to the C and G, this couple would have, or this fragment would have 18 bonds. So as you see, it would be much easier to separate this fragment when it has A and T, adenine and thymine, because it has less bonds. So now I hope that you would understand the difference between Tata box in eukaryotes or Pribnow box in prokaryotes, also this region called -10 region. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.